Hi, this is Gene Williams of Promise. Thanks for your interest in Promise. Happy to provide you with a Q2 update. I'll be making some forward-looking statements. I'd like to focus on some of the key events that we saw in Q2. We had a successful financing, extending our cash life into mid-2019. There was also a second positive phase 2 success in the amyloid field with BAN2401 in July, announced by Eisai and Biogen. In addition, our beta amyloid therapy, PMN310, has initiated cell line development, is moving full speed towards an IND filing and clinical trials in late 2019. And then lastly, our discovery platform is creating new selective antibodies against other toxic oligomer targets, like alpha-synuclein, and expanding our portfolio. What I'd like to do in this quarterly update is a slightly different focus than what we've done in the past. Three of these major events have generated significant investor questions, and I'd like to focus on trying to provide an answer to some of those questions that have come up, and hopefully that's useful for all of you listening. First, with the BAN2401, there was initial very strong positive response when they announced their data, but then some controversy arose when they talked about it in more detail at an Alzheimer's meeting. Many of our investors have asked us, well, how do you sort out that controversy? What's the perspective on BAN2401? So I'd like to address that. Secondly, we were very excited to initiate cell line development with a very high-quality cell line vendor, Celexis. But the question came up, why is that so important to the ultimate profitability and success of PMN310? And I'd like to address that question as well. And then thirdly, we have a discovery platform that has a unique capability to create highly selective antibodies for toxic oligomers. We've spoken quite a bit about how that is a unique competitive advantage for us in the field of neurodegenerative disease. And we often get the question about how we're applying that discovery platform to creating value, expanding our portfolio. We recently announced that we've made some progress against a new toxic oligomer for alpha-synuclein. So let me talk about that. What I'll do over the next few minutes is talk about those three things in a little bit more detail. We and others were very encouraged to see the second positive clinical trial result in the amyloid field with the announcement in July from Eisai and Biogen of the data from their BAN2401 study. For both BAN2401 and aducanumab, we saw very positive phase 2 signals. There was a strong dose-response curve. The higher the dose, the better response, and that's probably the most important thing you're looking for in a phase 2 study. In addition, there was a positive signal, positive separation from placebo on important clinical endpoints in the highest dose arm or arms. Both of these studies, we believe, were very positive phase 2 results. Wall Street saw it that way as well. When both were announced, there was a similar market value increase of roughly $15 billion. And when the BAN data came out, Wall Street analysts, to a very large extent, felt it's validated the amyloid hypothesis and raised the odds of approval for aducanumab. We agree. However, in the case of BAN, there was some controversy that came out after those trial results were first announced. It had to do with the fact that regulators suggested that fewer APOE4 patients be put in the highest dose arm because those patients have a higher risk of a side effect, ARIA-E. In addition, those patients are more likely to progress rapidly, so many felt that biased the study in favor of BAN2401, but there were two other factors that biased the study the other direction. In the second highest dose arm, the 10 mg arm, there were significantly more APOE4 carriers, but despite that handicap, it still showed a positive signal. And throughout the study, it appeared that the placebo progression was slower than what we've seen in other studies recently, like the aducanumab study and the ADNI study. So on net, the factors offset each other, and we feel it's a very positive phase 2 result. This is similar to the controversy that happened with aducanumab when they added 500 subjects. We believe that was widely misunderstood. If anything, that's probably positive news, because the variability that led Biogen to do that, which they'd done on prior studies in other neurodegenerative diseases, could have been due to either measurement error as you add more sites, or due to the drug working better than had been predicted. So on net, we feel that both these products have shown positive clinical signals in early stage studies, and most importantly, those positive signals are in line with the science. Now, I won't repeat all of the science that we disclosed, we shared a couple of weeks ago, but to summarize quickly, there are 4 forms of beta amyloid in the brain. Only one is toxic. So if you look at how selective different products are for that toxic oligomer, that helps explain the scientific results. what we've seen is that all of the failed studies bind monomer, and conversely, everything that binds monomer fails. Both aducanumab and BAN2401 are selected for aggregated forms of amyloid, including but not limited to the toxic oligomer. They both have a positive phase 2 signal. And Promise PMN310 is the most selected product of all for the toxic oligomer, which we think still positions us to be best in class. We see BAN2401 as a second very positive signal from a beta amyloid therapy and is encouraging for the field and raises the odds of both aducanumab success and our ultimate best-in-class position. Let me look at the second question that has come up for many investors, which is we announced cell line development, and many people say, why is that so important? Well, let me talk about what we've been doing over the last several months to maximize the value of PMN 310. In Q1, we focused on scientific differentiation. A reminder, PMN 310 was the best of over 300 candidates we identified using several epitopes, and it was the best at selectivity and functional benefit, which include preventing neurotoxicity and preventing propagation of toxic oligomers. Q1, we announced a great deal of scientific evidence suggesting PMN 310 might have superiority over competitive amyloid programs. We had no plaque binding with a lower risk of area E. We've recently extended that to looking at BAN2401 as well. We have that advantage over BAN as well. We have the greatest selectivity of any beta amyloid monoclonal antibody that we're aware of, and we had a stronger binding signal to the brain homogenate that was concentrated to include the toxic oligomers as compared to aducanumab and other programs that have been in the clinic. Q2, we've been focusing on other aspects of maximizing the value of PMN 310, initiating manufacturing, which is the critical step for getting to a regulatory approval to begin clinical trials, and we're on track to do that by the end of 2019. But also we try to select manufacturing vendors who can give us a very high yield process. Manufacturing cost, we expect pricing to be more challenging going forward, and we think lower pricing makes a great deal of sense in the Alzheimer's market to enable broader utilization. So we think cost of goods will be critical in driving profitability. Let me show you a little bit more on that front. Now, manufacturing is integral to the product and it impacts NPV. The FDA considers manufacturing to be an important part of what your product is. While it's theoretically possible to change your cell line in the process once you're in the clinic, that's very, very challenging to do. You don't want to do that. So if we look at the expected NPV analysis at the end of phase 2, Evaluate Pharma did something that was published a couple of years ago showing about a $10 billion NPV, expected NPV, adjusting for risk. And we commissioned an analysis from the next big pharma analyst who looked at these sorts of issues for in-licensing candidates at large pharma and came up with a very similar number, $12 billion. We assumed a $10,000 annual price, 40% market share of symptomatic treated patients, and an annual cost of goods of $4,500. Now, those expected NPVs are very much in line with the market value increases that we showed before with successful phase 2 data. Both aducanumab and BAN2401 generated market cap increases in the $10 to $15 billion range. But look at how sensitive that NPV analysis is for cost of goods. If the cost of goods can go as low as $3,000 a year, which could come from a more productive cell line, that could increase the NPV to $18 billion, expected NPV, adjusting for risk at the end of phase 2. If it's a less productive cell line and the cost of goods is $6,000, that can cut it in half to $6 billion. So we're working very hard on finding a very, very high productivity, high-yielding cell line, and that's what we're very excited to be moving forward on right now as we try to maximize the value. Now, these are the sorts of analyses that large pharma partners would be doing in a licensing or an acquisition deal in a couple of years, or next year or beyond when we're in the clinic, and therefore this is very important to their calculation of overall value. Let me turn to the last question, which is what are our plans for the discovery platform and expanding our portfolio? Talk a little bit about the neurodegenerative space, though, just some comparable or comparative analyses. There are 4 main targets that we're focusing on for toxic oligomer monoclonal antibodies that impact the root cause of neurodegenerative disease. Amyloid beta and tau, the first 2 rows, are the 2 toxic oligomers that kill neurons in Alzheimer's. And then TDP-43 and alpha-synuclein down below are other important toxic oligomer targets. TDP-43 in ALS and frontotemporal lobe dementia, and then alpha-synuclein in Parkinson's and Lewy body dementia. We've talked already about the market value increases of large pharma companies with phase 2 success in beta amyloid. Uh, we have a number of examples, though, recently of partnering deals, and we've done a more extensive analysis. These are just some representative examples. In the tau field, Prothena sold an asset to Celgene. It was preclinical, $50 million upfront and $700 million downstream potential milestones. Similarly, Prothena Celgene's deal for TDP-43 had the same economics, $50 million upfront, $700 million downstream. And in the alpha-synuclein field, BioArctic, the same company that invented BAN2401, licensed their alpha-synuclein antibody to AbbVie for $60 million upfront with $675 million downstream. PROMIS is making progress on each one of these fronts, and we expect within a few months to have data showing that selective antibodies are functionally beneficial and provide other information that may be very attractive to large pharma partners. We're on track for initiating our clinical trial with beta amyloid in late 2019. We have a design using biomarkers that could show some potential biologic benefit in those patients by the middle of 2020. For tau, the epitope identification is in its final stages, and we expect results very, very soon. For TDP-43, we've announced already some initial data with selective antibodies. We have several epitope targets and are looking at candidates in the coming weeks and months. And with alpha-synuclein, we've been making great progress. We announced previously we have identified 5 epitope targets. We have over 100 antibodies that show selectivity, and we're looking for the best one in terms of functional benefit right now. So in summary, I think Q2 has been another quarter of tremendous progress for us at PROMIS. We had a successful financing, which has centered our cash life and has enabled us to keep expanding our portfolio and advancing our lead program, PMN310. BAN2401 has provided greater validation of our science perspective on amyloid, that selectivity leads to clinical success. The more selective you are for aggregated forms, particularly the toxic oligomer, the better your clinical benefit and the greater the chance of being best in class. PMN310, which we believe will be the best in class antibody in amyloid, is moving forward to an IND and clinical trial, and we've been making great progress on coming up with high yield, highly productive cell lines that can optimize the profitability of the product going forward. And our discovery platform is now moving full speed ahead on other targets. Now that PMN310 is moving rapidly toward the clinic, we're moving full speed ahead to expand our portfolio into other targets that are of great interest to large pharma partners, and that should give us a growing number of partnering options over the coming months. Thanks very much for your interest in PROMIS.